That's what I was doing.
If somebody would remind me tomorrow in lab, we need to have you start another broth culture on Wednesday or Friday, because you're going to do a motility test, and you would like a culture no more than 48 hours to do the motility test. So I'll try to remind you, but help me.
What did you do? Does he have Are you serious? Did he have a girlfriend? Yeah, he did. No, he's just that color. And for like 2 seconds. I think it did. No, I'm not going to. Okay.
Okay, let's do some metabolism, because this is going to be relevant to some upcoming labs, particularly after the first lab exam, when we start doing some diagnostic information about your unknown that doesn't depend upon the structural staining, but instead depends upon an analysis of what kind of enzymes does your bacteria have, what kind of products does it produce from a metabolic reaction. So this will be kind of introductory material for down the road for lab a couple of weeks. So what does the term metabolism refer to?
How fast your body or something can convert your energy source?
That is the kind of popular vernacular, idea of metabolism. It has to do with kind of rate of burning fuel, for example. But biochemically speaking, what's metabolism referred to, John?
All the chemical reactions in the body.
All the chemical reactions in the body. It is the totality of all of the chemical reactions in the body. And we typically subdivide that big bucket of all the chemical reactions into 2 subcategories, catabolism and anabolism. What are the differences in the two? What's catabolism? Breaking down things. Okay? The way that I think it's easy to remember it is, you probably all heard of anabolic steroids before. What do anabolic steroids do? They build muscle mass. Okay? So these are the synthetic reactions. And these are the reactions that break down molecules. Okay? Typically, catabolic reactions, are they oxidation or reduction reactions? Are the substrates oxidized or reduced? Glucose is converted into carbon dioxide and water by a complex set of catabolic reactions we call cellular respiration. During cellular respiration, is glucose oxidized or reduced? It is oxidized. Hydrogens are removed from the glucose molecule, and eventually we're left with carbon dioxide. So these tend to be oxidizing the substrate. What about anabolic reactions? We tend to oxidize or reduce the substrates when we have anabolic reactions. We reduce. Okay? So these tend to be reduction substrates. Are catabolic reactions exergonic or endergonic? Remember those terms? They tend to be exergonic reactions. And the energy that is liberated from these is used to do what? Drive the endergonic synthesis of ATP. Anabolic reactions tend to be endergonic reactions. They require an input. of ATP to happen. So what's the relationship between catabolic and anabolic reactions?
They're opposites.
They're opposites, and oftentimes they are coupled together. And so we use a catabolic reaction to generate ATP to then drive the endergonic synthesis of new molecules. And then what can those new molecules later be used for? They can be catabolically broken down to make more ATP. So these reactions are interdependent and oftentimes coupled with one another. We're going to be dealing with primarily catabolic reactions, and particularly in the lab context. But it is important to understand that metabolism as a whole does have this dual role. One role is to generate ATP, but the other role is to produce what we refer to as carbon skeletons that can be used in the synthesis of new molecules. So take a look at this overhead up here. This is kind of a summary, a gross overview summary of metabolism. We can, for example, take molecules like carbohydrates, glucose, and convert them through glycolysis, Krebs cycle, and electron transport system to produce ATP. That's kind of the catabolic role of cellular respiration. But also what can happen in cellular respiration is instead of completely breaking down glucose into carbon dioxide and water, we can partially break it down. And we can funnel off some of these intermediate compounds of glycolysis or the Krebs cycle and use those as building blocks, carbon skeletons, for biosynthetic reactions. Okay, so here, acetyl-CoA, which is produced following glycolysis right before you enter the Krebs cycle, the cell can take this compound and completely break it down by the Krebs cycle, and we get the maximum amount of energy out of it. But biochemically, the cell can also take acetyl-CoA and use it to synthesize fatty acids, which are used in fat production. Okay? Compounds that are part of the Krebs cycle can be converted into amino acids, just as well as amino acids can be converted into compounds of the Krebs cycle. So there's this interesting relationship between the anabolic role of cellular respiration and the catabolic role. In cell biology and most other courses, we characterize organisms into two nutritional classes. Remember what those two nutritional classes are? Autotroph and heterotroph. Okay? And we kind of lie to you a little bit in doing that. Actually, we're oversimplifying things. Because basically what we say is that heterotrophs are the consumers and autotrophs are the producers, right? So plants are autotrophs, we're heterotrophs. Okay? In the microbial world, things are a little bit more complicated than that. What we do to characterize the metabolism of bacteria is to look at two factors. What is the energy source that the microorganisms are using? And what is the carbon source that they're using? If organisms obtain their carbon from organic sources, like glucose, then those organisms are said to be heterotrophs. If they obtain their carbon from inorganic sources, what's an inorganic source of carbon?
CO2.
CO2. Then they are an autotroph. This really technically means nothing about photosynthesis. All this term autotroph refers to is where you're getting your carbon you use in metabolism. It means you're getting your carbon from an inorganic source. Here, if you're a heterotroph, you're getting your carbon from an organic source. There are two possible sources of energy. One is chemical compounds. If you get your energy from chemical compounds, you are a chemotroph. Most of the chemical compounds are organic in nature, like glucose. But in the bacterial world, inorganic molecules can also be used as an energy source. That's kind of unusual. What's another potential source of energy besides chemical compounds that microorganisms could use? Sunlight. If you produce your ATP by using sunlight energy, you're a phototroph. If you produce your ATP by using chemical compounds as a source of energy, you're a chemotroph. So to really characterize an organism in terms of its nutritional strategy, we need to apply two names to it. One that indicates its source of energy, and one that indicates its source of carbon. So how many combinations are there? There's 4 possible combinations. And what's interesting is that in the microbial world, all 4 nutritional strategies are represented. When you get down to eukaryotic organisms, only 2 of those strategies are represented. What are the 2 eukaryotic strategies that are represented? Photoautotrophy. Energy from the sun, carbon from CO2. Green plants are photoautotrophs, which we just cheat and call autotrophs. Okay. What is the other strategy that's represented in eukaryotic organisms? The chemoheterotrophs. You use organic molecules like glucose as a source of energy, and you also use organic molecules like glucose as a source of carbon. Now, both of these are also represented in the microbial world. We talked about organisms that are photoautotrophic, and they included both organisms that carry out oxygenic photosynthesis, but they also include organisms that carry out the anoxygenic photosynthesis. Remember from last exam which group of organisms carry out the oxygenic photosynthesis? The photoautotrophs, and they produce O2. There were those stromatolites that I showed you from Shark Bay, some of the first photosynthetic organisms. Those were the cyanobacteria. Okay, so they're represented in this group. But then we also talked about the little variation on photosynthesis, where the green and purple sulfur bacteria could break down things like hydrogen sulfide as a source of electrons instead of water. They're also photoautotrophs. Okay? We will not have any occasion to deal with any of these photoautotrophic organisms in lab. But they are very important organisms in terms of the functioning of our whole world. They are the producer organisms. They're the ones that convert sunlight energy into chemical energy. We are chemoheterotrophs. Okay? We get our energy from carbon, and we get our carbon skeletons from organic carbon. Most bacteria are also chemoheterotrophs, including all of your unknowns. Because what they're doing is they're living on the nutrients in agar. They're using amino acids as a source of carbon and nitrogen, and also as a source of energy. But there are two other patterns which are only represented in the microbial world. Photoheterotrophs, interesting group of organisms. Where do they get their carbon? From organic molecules. Well, where do they get their energy? From the sun. Okay. These are some organisms we'll talk about a little bit later on. The green non-sulfur bacteria and the purple non-sulfur bacteria. This is a very interesting strategy if you think about it, though. Why would you use an organic source of carbon, but then use sunlight instead of those same organic molecules for energy? It's a different niche. Okay? Microorganisms have evolved to really fill any imaginable niche that's available. And then there are these guys. These are really interesting. The chemoautotrophs. Because these chemoautotrophs use CO2 as their source of carbon, which is inorganic. They use molecules, chemical compounds, as their source of energy. But the chemical compounds they use are inorganic in nature. Sometimes these chemoautotrophs are referred to as lithotrophs. These are literally the rock-eating bacteria. They take inorganic molecules and use them as their source of energy. And they also use inorganic molecules as their source of carbon. So another place we lie to you, or simplify things, I do it in cell biology all the time, is I say that all organisms rely either directly or indirectly on what to survive? The sun, right? Because organisms that use sun energy and turn it into chemical energy are the producers, and then the consumers eat the producers. These lithotrophs, these chemoautotrophs, are exceptions to that rule. Do they need sun energy? Nope, they can use inorganic molecules as their source of energy and inorganic molecules as their carbon source. So the idea that if the sun were to go out permanently, everything on the world would die, almost everything, except the chemoautotrophic bacteria. So we'll talk a little bit more about some of these strategies. Basically, we're going to take these groups, and we're going to talk first about chemoheterotrophs and spend most of our time talking about that. We'll talk a little bit more about chemoautotrophy, then talk about photoautotrophs, and then say just a little tiny bit about photoheterotrophs. But this is what we're going to spend most of our time on, because this is what is going to relate to lab and our lab tests that we're going to do on our unknown. So when we talk about chemoheterotrophs, really what we need to delve into is aerobic respiration, anaerobic respiration, and fermentation. Because it's these pathways that really produce the energy and the carbon skeletons that are needed for biosynthetic reactions. Now, I got a little bit ahead of myself last time doing a little bit of review of aerobic cellular respiration. That's fine. Obviously, you need to remember some very fundamental things about aerobic cellular respiration that we talked about last time. And that is that the process involves the sequences of glycolysis, Krebs cycle, electron transport, that it's pretty efficient in making ATP. Per glucose, the maximum number of ATP you can get is 38 ATP. The real energy payoff for ATP production comes from the electron transport chain. Because remember, that's where the reduced coenzymes are recycled. And as that recycling happens and electrons are being passed through that electron transport chain, what's set up across that membrane is electron transport first. Hydrogen ion gradient. Okay, high hydrogen ions at one side, low at another. And as those then flow through the membrane through that ATPase channel protein, we can make ATP. Do you remember the equivalency for every reduced NAD that donates its electrons to the electron transport chain? How many ATP can you get out? 3. For each reduced FAD, 2. A little bit of ATP is produced directly in both glycolysis and during the Krebs cycle by substrate level phosphorylation. Remember that term? Whereas the ATP that's produced during electron transport is produced by oxidative phosphorylation. Okay. So those are some of the highlights. We produce a lot of CO2 as a result of aerobic cellular respiration as well. One thing I would like to mention right now that will be relevant to lab later on has to do with the electron transport chain and the quote-unquote carrier molecules that make up that particular sequence. Remember, there are a bunch of molecules in the electron transport chain that can donate and accept hydrogens, kind of pass them down the chain toward oxygen. Several of those are iron-containing proteins. Anybody remember what the iron-containing proteins are of the electron transport chain? What they're called? Kind of similar to hemoglobin. Hemoglobin's an iron-containing proteins. Cytochromes. In fact, an old name for the electron transport chain was the cytochrome system. Don't use that so much. Now, not all of these carriers are cytochromes, but several are. And some species use a particular cytochrome at the very end of the electron transport chain, called cytochrome A3. And not all species do this. Ooh, opportunity to distinguish between species. Okay? There is a fairly simple test that we can do to determine whether your unknown has cytochrome A3 in its electron transport chain. And that particular test is a test that we will do called the oxidase test. And that can be really important in starting to differentiate between different kinds of unknown organisms. So we'll come back and do that later on. We also then talked about anaerobic respiration and the differences between aerobic and anaerobic. The similarities are that they share the same three set of chemical reactions: glycolysis, Krebs cycle, electron transfer. What's the difference?
The final electron.
The final electron acceptor is not O2, as it is in aerobic. It is some inorganic molecule that contains oxygen, typically, like sulfate or nitrate or carbonate. And those compounds pick up the electrons from the electron transport chain and become reduced. So sulfate can be converted into sulfite. Nitrate can be converted into nitrite. And we'll talk a little bit more about that later on. Is that as energy efficient as aerobic? No. Wide range of energy produced, they just tell you here, somewhere in the range of 2 to 36 ATP. But less than aerobic. So the consequences are that if you've got two things, if you've got an organism that can do both, what do we call those? Facultative. Okay, if we've got a facultative anaerobic, where is it going to grow better? Aerobic conditions or anaerobic? Aerobic. That's what we use our gas pack for tomorrow. We'll grow your unknowns in an anaerobic environment and see, number one, do they grow? And two, then, how do they grow in the gas pack compared to how they grow in the normal incubator? So that'll be real important information for a lot of you. Now, typically in anaerobic, the final electron acceptor is inorganic. But some microorganisms are also able to use organic molecules in anaerobic respiration. Any of you like to eat fish? Okay. How do you know when fish has gone bad? It's really got a very characteristic smell, doesn't it? Fresh caught fish does not have that fishy smell. Well, as soon as a fish dies, bacteria that are resident populations within that fish start to break down the tissue. And when they begin to catabolize the protein, they produce a byproduct, a nitrogen-containing byproduct called TMAO. Trimethylamine oxide. So that's what makes a fishy smell, right? No, not actually. That's an odorless compound. Other bacteria, though, in the fish can use TMAO as the final electron acceptor in anaerobic respiration. And what happens to this compound? it becomes reduced. It picks up electrons from the electron transport chain. And its reduced form, this smells. The human nose is really, really sensitive to reduced TMAO. You can literally smell a few molecules of it. So as more bacteria begin to break the protein in the fish down, and then other bacteria then reduce that by anaerobic respiration, we get more and more of this produced. It starts smelling worse and worse and worse, which is indicative of microbial growth. So at some point in time you go, can't eat that, toss it away. And that's a good choice, okay? Because now it's pretty heavily contaminated with bacteria. Fermentation. Fermentation consists of glycolysis and then some additional step which will regenerate the oxidized forms of NAD. So glycolysis plus a recycling step. No Krebs cycle, no electron transport. Poor energy production, 2 ATP per glucose. What's kind of interesting is that microorganisms are really adaptable. And if you look at the growth rates of facultative organisms that can either carry out aerobic respiration if oxygen is present, or fermentation if it's absent, some of those facultative can grow just as fast in the absence of oxygen as they do the presence of oxygen. And wow, that's kind of unusual, because aerobic is so much more efficient at producing ATP. How do they do that? They really incredibly speed up the rate of fermentation in the absence of oxygen. That particular response is called the Pasteur effect. Louis Pasteur first observed that. He goes, wow, how do these fermentative organisms grow so fast? They just ferment really, really fast. In fermentation, there has to be some place for the electrons to go that were first removed from glucose. Where do they go when they're removed from glucose in fermentation? Remember? In glycolysis? What picks up those electrons that are stripped off of glucose? We go from glucose to 2-pyruvate. We lose some electrons in the process. What picks them up? Oxidized NAD. Okay? So when reduced NAD is reoxidized again, where do those electrons go? There's got to be some electron acceptor, right? Well, in aerobic, it's oxygen. In anaerobic, it's probably nitrate or something like that. But what kind of molecule is the electron acceptor in fermentation? What defines fermentation? The fact that that electron acceptor is an organic molecule. So fermentation is characterized by having an organic electron acceptor. There are a variety of different organic electron acceptors that can be used in fermentation depending upon the species of microorganism and depending upon the enzymes that they have. So different species produce different end products from fermenting even the same sugar. There are a variety of acids that can be produced by the fermentation process. But there are also alcohols that can be produced. There may be gases that are produced as well during fermentation. Carbon dioxide, hydrogen. Remember, this is really pretty much a consequence of what enzymes you have. And enzymes are determined by what genes you have. So this gives us an interesting way to kind of differentiate among different species of microorganisms by looking at their fermentative end products, therefore understanding something about the enzymes that they have. And different species of organisms have different enzymes because they have different genes. So this can be really important diagnostic information. So, here are a couple of fermentative strategies that you are probably familiar with already. Okay, whoops. So here's glucose, and glucose is converted to pyruvic acid, NAD is reduced at that particular point. So these particular reactions are just involved in recycling the reduced NAD back to the oxidized form again. Okay? So, in lactic acid fermentation, pyruvate is converted into lactic acid. This is a 3-carbon compound. This is a 3-carbon compound. So does this kind of fermentation produce any CO2? No. Hasn't lost any carbon. Lactic acid, however, is acidic. And as these organisms ferment, they lower the pH of their environment. And that can have an effect on other molecules in their environment, like milk protein casein in dairy products. Lactic acid fermenting bacteria, unless the milk has been sterilized, are there. Pasteurization doesn't kill those lactic acid fermenting bacteria. Now, if you keep your milk refrigerated, they're going to grow pretty slow. But over a period of time, they will begin to ferment the sugar, lactose. They'll produce lactic acid. That will lower the pH, and eventually the milk protein will curdle, and the milk will become sour. Okay, even in the refrigerator. You've got to have the right enzymes to be able to do this. Do we have this enzyme as well? Can we do lactic acid fermentation? Yep. That's what happens when we overexert and exercise, and the circulatory system can't provide enough oxygen to our muscle cells. So they switch over temporarily to fermentative pathways to keep cranking out a little bit of energy, and lactic acid accumulates for muscles. Another strategy that yeast and some microorganisms can employ, because they have different enzymes, is to first convert pyruvate into acetaldehyde. 3 carbons here, 2 carbons here. We evolve CO2. That's where that carbon is lost. Acetaldehyde is then reduced into ethyl alcohol. Can we do this? Nope. Otherwise, long distance running would be an interesting proposition, right? Stagger across the finish line. This is an example of some of the different types of outcomes that you can get from different microorganisms by different fermentative pathways. So in Clostridium, pyruvate can be reduced into butyric acid. You know what butyric acid is? It gives the rancid smell associated with spoiled butter. Have you ever smelled a ginkgo tree's fruits? I don't know what a ginkgo tree looks like. Nasty smelling. There's butyric acid in the seed coat of those female seeds of the ginkgo tree. And they fall on the sidewalk and you step on them and they just smell horrible. We used to have one over by Sherman. There used to be one out here on the wall too. Proteus bacteria ferment and produce formic acid plus CO2 and hydrogen. Why? Because they have different enzymes than Clostridium. Propionobacteria produce several acids, including propionic acid. These are the bacteria that are used in Swiss cheese production. And propionic acid has a characteristic odor as well, the odor you associate with Swiss cheese. Some bacteria produce acetic acid. Some organisms produce a variety of different acidic compounds, a mixture of them. This is one we're going to talk about later on. We're going to do in lab. Members of the genus Enterobacter, which are really, really hard to distinguish from Escherichia. They're kind of cousins, a lot alike. But one of the ways you can really distinguish between those is when pyruvate is fermented by Escherichia, E. coli, it does this mixed acid fermentation. And there's a way you can detect that. Enterobacter, on the other hand, produces this compound called acetylmethylcarbinol, which is later then reduced into 2,3-butadiol. And there's a test we can use for this acetylmethylcarbinol. It's called the Voges-Proskauer test. We'll do that one too. So that's really important to distinguish between these two general. This one's hard to see, but this just shows you again some of the kind of products that can be produced from fermentation. Alcohols, isopropanol. Again, butyric acid, fumaric acid. There's a whole bunch of different kinds of outcomes of fermentation. So what are the general categories or types of fermentation? Fermentation that generates alcohols. Ethanol, butanol, isopropanol. The one I just mentioned, the 2,3-butadiol fermentation, which is derived from acetylmethylcarbinol. We'll detect that later by doing the Voges-Proskauer test. So we'll come back and see that in a little bit. A lot of fermentation produces acids. Lactic acid fermentation is pretty common. If you only produce lactic acid, then that's referred to as homolactic fermentation. Heterolactic fermentation involves producing lactic acid plus some other acidic compound, more than likely acetic acid. Mixed acid fermentation is the result of producing a whole bunch of different compounds. That's oftentimes accompanied by the production of gas. This kind of fermentation really dramatically lowers the pH of the environment. This mixed acid fermentation is associated with really a lot of accumulation of acid. And there is a test that we can do and will do in lab that will detect mixed acid fermentation. It's called the methyl red test. One of the first fermentation tests that we'll do is a battery of tests called the phenol red fermentation tests. This test consists of a broth, like tryptic soy agar, to which a sugar has been added. Glucose, sucrose, lactose, maltose, some sugar that we want to see if the organism can ferment it. There is a little small inverted tube placed in this tube of broth. That little inverted tube is called a Durham tube. And then also mixed into the broth is phenol red. What's the usefulness of phenol red? Have you ever used phenol red before?
It's a pH indicator.
It is a pH indicator. Litmus is a pH indicator. You might use litmus. If the pH is greater than 6.9, then phenol red is red. If it's less than pH 6.9, it's yellow. So where we'll pick up Thursday is talk a little bit about these fermentation tests and how you can determine some key information, because we will be doing these fermentation tests right after the lab exam. Pardon me?
When is that?
It's coming up in a couple of weeks. It's unpublished. No, we got a couple of weeks worth of lab work.
Okay, wow.
It's the Wednesday after fall break. 22nd. Oh, I looked at the syllabus. Does anybody know when the next exam is coming up? The lecture exam? 22nd. Probably not. Probably not that far away.